Professor Dr. Laure sir for giving me this opportunity. Because from my introduction, you must have realized that since my PhD days, I'm working with secondary metabolites, and it is a topic which is really close to my heart. So without wasting any further time, let us now jump into the presentation. So one thing is that in any case, in any slide, if you have any doubt, you can stop me at that time and get it clarified. Or otherwise, at the end of the presentation also, you can ask me your doubt. Okay, so is it visible, my presentation slide?
Yes.
Yes, ma'am.
Okay, thank you so much. So for the time being, I'm going to shut off my video also in order to preserve the bandwidth. And afterwards, during the question answer session, I will again switch it on. If that is all right. Yes. So today I will be talking about the secondary metabolite production in plant cell cultures under in vitro conditions. Now, as you will realize as I'm moving ahead into the presentation, it's a really vast topic. And so I have decided to give you just a basic glimpse about the topic. And there are n number of review articles that are available on each of these aspects. And separate books also have been published. So this is, we can say, a small attempt in understanding this vast topic. So as all of us know, since you are doing your PhD, you are aware of these things. But as always, I think that it's best to have a recapitulation of the knowledge that we are having. So why the plant cell culture is so very much successful? All of us have heard about animal cell culture also. But obviously there are a number of limitations in animal cell culture. But on the other hand, we have very many success stories from plant cell culture. So one of the most important properties that has helped us in this high success rate is the property of totipotency in case of plant cells. The totipotency is a property which allows a single cell to be regenerated into a whole plant. That is why plant tissue culture, even at a commercial level, is so very much successful. Then all the plant cells show the property of autonomy. That means the cells are able to grow away from the mother plant. So that's why we can culture them in glass vessels, and we can grow them in artificial nutrient media with controlled environmental conditions. Third important property is the high degree of plasticity that is shown by the plants. That means, as per our wish, we can change the developmental program that is there, and we can decide that, let's say, from a leaf explant, whether I want to induce a root or a shoot or a callus and so on and so forth. So that is what is called as plasticity. So like a plastic can be molded into various shapes, we can mold the plant cells into any kind of a developmental pathway that we are interested in. So that is the meaning of this plasticity term. And then plant cells can be made competent with modifications in media and environment. I'm sure all of you are quite aware of these things. So that is why the plant tissue culture and plant cell culture industry also has been highly successful due to all of these factors. So let us move ahead. I am directly now jumping into the various types of tissue cultures that are there, and I will be concentrating mostly on the callus culture and the cell culture, because these are the two techniques which have given us the maximum important secondary metabolites that are being produced under in vitro conditions. There are many other types of culture types that have given, like in some cases you have got shoot cultures, in some cases you have got root cultures. But for this today's talk, I will be talking only mostly about the cell cultures and a little bit about the callus cultures. Okay, so all these others are just the different types of culture methodologies that you can use when you want to do any kind of in vitro related studies. Again, a brief single slide overview of all the basic PTC requirements. Here, as you know, on the left hand side, I have just enlisted the important factors that will help you in getting a good results in your tissue culture. I'm not going to discuss each of these in detail. But my most important point is related to this selection of biological material. So as you know, in micropropagation, there is something called as a stage zero. So even before you begin your tissue culture experiments or cell culture experiments, you have to choose a nice or an elite mother plant, because that is the most important requirement for success of your culture. So I will give you one example from my PhD studies. As you heard, I worked on Taxus baccata cell culture. So Taxol is an important secondary metabolite which is used worldwide. Now it is a well-known drug also, which is effective against breast and ovarian cancer. So aim of my PhD work was to increase the production of Taxol in cell culture. So I screened around 30 mother plants of the Taxa species, and as expected, the mother plant that was already having a decent amount of Taxol in it, I was able to increase the Taxol content subsequently in the cell culture medium by media manipulations and environmental conditions manipulations up to 300 times from the intact plant. So actually, the US patent was based on that media formulation, which gave me this super high yield of Taxol under in vitro conditions. So I want to emphasize the importance of biological material. So when you are starting with your tissue culture experiments, spend enough amount of time in choosing the correct plant, because it will give you better results. Just do not go out into the backyard and choose whatever plant is available in the closest vicinity. Spend some efforts in selecting a better or an elite mother plant, as it is called in technical terms. Then obviously all the other factors you will have to manipulate in order to get the best results out of the best biological material. मराठीमध्ये ह्याला एक खूप चांगली म्हण आहे की आडातच नाही तर पोहऱ्यात कुठून येणार. जर का तुमचं मदर प्लांटच चांगल्या दर्जाचं नसेल, तर तुम्ही इकडे पुढे टिश्यू कल्चरमध्ये कितीही डोकेफोड केली तरी तुम्हाला डिझायरेबल रिझल्ट्स मिळणार नाहीत. सो ही गोष्ट कायम लक्षात ठेवायची. So the selection of a suitable or an elite biological material is extremely important. And then obviously you can go on working on the nutritional medium, the balance of growth hormones, then the environmental condition modifications, and so on and so forth. Now, all of you know that plants require a number of secondary metabolites which are not directly related to their primary metabolism, but they are essential in the physiology, defense, and adaptation to the environment. So, all of these are grouped under the umbrella term of secondary metabolites, and you are aware that they are subgrouped into these various classes like terpenes, steroids, phenolics, flavonoids, and alkaloids. Now, why I have written these? Because from each of these subclasses of secondary metabolites, we have got some very star drug molecules that are already available in the market, and that's why you cannot say ki steroids are not important or phenolics are not important, only alkaloids are important. So there is nothing like that. Each of these secondary metabolites have really helped us to fight a number of diseases. So around 2 lakh of such important molecules have been identified, which have shown either a therapeutic property or a nutraceutical property or a cosmeceutical property. So therapeutic property, all of you understand, it is directly related to the medicinal activity of the plant. Now these two terms, the nutraceuticals and the cosmeceuticals, they are what you call as sunrise fields. या fields ना पुढे जाऊन खूप महत्व येणार आहे. So nutraceutical means, as you can see, nutrition and pharmaceuticals. It's a combination of two terms. So the food items that you eat, if they are going to give you a therapeutic benefit, or if they are going to give you any type of a medicinal benefit, that category is called as a nutraceutical. So all of our spices fall into this category. So they are essential part of our daily diet, and also they are exhibiting a lot of therapeutic properties. Especially during COVID outbreak, we have seen how the spices have played an important role during that time period. Similarly, cosmeceuticals is the category wherein you have a cosmetic plus a pharmaceutical. So we have so many examples in Ayurveda related to that, like Chandan is there, Haldi is there, and you yourself can think of many such metabolites which have both cosmetic as well as therapeutic properties related to them. So, like turmeric has a skin whitening effect as well as it has antimicrobial effects. So combination of these two properties. Similarly, chandan or sandalwood, as it is called, it has got a cooling effect as well as a wrinkle smoothing effect. So such types of two activities exhibiting stuff is called as a cosmeceutical. So this is very, very active. These two fields are really very active, and people, many industries, in fact, especially in countries like China, Korea, and Japan, are very much interested in these areas. Now, this diagram all of you must have seen during your MSc days, so I'm not going to spend much time over here. It is just to give you an overview, okay, how intimately the primary and the secondary metabolism is related to each other. And from the pathways of primary metabolites, we have the branches coming out which are going to give rise to these different classes of secondary metabolites, which are shown here in purple boxes. Now, I have tried to make a list of the various groups, as I was telling you, okay, which have some commercial importance. Because whenever you say production of secondary metabolites in vitro, you have a large commercial angle to it. Because a large number of companies are into this field, and wherever we are getting good money out of this work, people are very much interested in scaling up that work. So it's not only lab related research, but it has to be translated at a commercial level. So as you can see over here, I'm not going to read the entire thing, but you have so many categories wherein we have so many examples where people have already standardized the production mechanisms of these. But again, as I keep on telling that, people ask me ki, oh, but already so much work has been done, then why you need to work? So one best answer to that is given in the case of antibiotics example. So people were thinking that, oh, we have got so many antibiotics, now what is the need for doing any kind of an antibiotics research? But you see that the prokaryotic pathogens evolve themselves so continuously that they are developing resistance against many antibiotics, and many antibiotics have now become useless. So continuously we have to keep on searching for newer and newer antibiotic molecules, now from marine organisms, from desert plants, plants growing in highly stressful environments. So the search is endless, that is what I mean to tell you. So there is nothing like ki so many people have worked on it earlier, then why we should now work on it? Because there is a continuous evolution of the pathogenic organisms. We have to keep on searching for newer and newer molecules. Also for categories such as enzymes, etc. Again, we want to increase the efficiency of enzyme action. For pigments, now again, I would like to emphasize this area. Somehow in India, this area is very much neglected. Again, in Japan, there is a big industry wherein edible pigments and their production in bioreactors is a very big industry. So they are continuously extracting pigments, edible pigments from plant and microbe sources. And there is a huge demand for these edible pigments. Now, nobody over there uses any of the synthetic chemicals for giving colors to their foods and bakery products. Everything is organic, everything is from plant or microbial sources. So this again is a very very as I like to call it, a sunrise area, wherein lot of plants from our own biodiversity can be exploited for better uses. This is an example from our very own CFTRI, Central Food Technological Research Institute, which is situated at Mysore. So I had visited that lab during my tenure in NCL, and these are the various beet cell culture lines that they have established. So the initially the callus was derived from the roots, and by manipulating the culture conditions, so here I have put only a limited slide wherein only variations in the red pigment colors are seen. But they have been working on this beetroot callus culture for last more than 15 years, and they have now got a whole spectrum of colors. Everything right from pale yellow to the deepest maroonish red. They have been able to get these different colored pigments via cell culture, and last I heard, they were in talks with a industry in order to transfer this technology. So they have even scaled it up to a bioreactor level, and they are now looking at commercial exploitation of this, because as you know, beet gives edible pigment, so it can be without any worries of cytotoxicity or carcinogenicity. These pigments can be happily used even in cosmetic products. So not only food products, even in cosmetic products, these pigments have a huge demand. So I will not go into the details of this slide, because callus cultures, I am not going into too many details about it. So moving ahead with the cell culture. So as I told you, these are the so many different areas, as I have already mentioned in the earlier slide, where people are actively working in order to find more and more effective secondary metabolites. Then the advantages of using in vitro cell cultures. So again, as I keep on telling to my MSc students, is that you need to have a very good logic why you want to start basically any type of a tissue culture experiment. So one of the reasons can be that your product has to be very very climate friendly, because as you know, currently that has got a very high impact, because many of the consumers are also becoming aware of these, that while production of these entities, any kind of climate damage has not happened. So all these things are now many of the consumers are quite aware of. So that is one thing, that you want to produce these secondary metabolites in a climate friendly manner. So tissue culture is one of the best ways in which you can do that. Secondly, if your secondary metabolite is highly expensive, like again going back to the example of Taxol. Taxol is an extremely expensive molecule if you want to produce it from live plants. So, in order to just get 1 milligram of Taxol from native trees, you have to have bark of more than 10 trees that needs to be stripped off and then extracted to make Taxol. So, it is really very destructive for the environment. But in tissue culture, you can use a number of methodologies in order to improve the production of that compound, and all the efforts that are you are going to put into the tissue culture are worth it if the compound being produced is going to give you a very good commercial value out of it, right? So that is, those are two or three logics that you have to apply. If you do not use this logic, then down the line you might end up in getting many more questions being raised for your work. Then another important point is that you can do the improvement in many fold, like multiple folds. So 100-fold, 200-fold, 300-fold improvement in secondary metabolite production is not possible by your normal agricultural methodologies. But tissue culture experiments, in vitro manipulations can really improve the yield of the compound to a great extent. And then when you apply the bioreactor level technology, which many companies all over the world are applying routinely, and all those products are already available in the market, then that becomes a very economical proposition as well. Okay. Then again, I have not covered in this talk anything much related to the entire transformation technology. So, agrobacterium mediated transformation, either through production of hairy roots or through tumors, it has again given a huge number of boost to the entire area of secondary metabolite production. But because of the paucity of time, I'm not going to cover that aspect in detail in my talk. So that is another interesting feature that can be thought of when you're using tissue culture. So manipulating culture media, choosing the best elite mother plant, manipulating the environmental conditions. So, all of these things are happening on untransformed cell cultures, for which I'm going to give lot many examples, and my subsequent slides are based on that. But there is this another important area of transformed tissue cultures, wherein you use a number of methodologies like direct gene delivery or agrobacterium mediated transformations in order to transfer actually one or two important genes into your plant cultures in order to improve the production of the metabolites. So that is coming under the purview of the genetic engineering, and many separate books and reviews are available related to that topic. So that is possible to do only if you have standardized your tissue culture system, like, right? So the non-transformed tissue culture system needs to be in place before you can go in for your genetic engineering experiments. And then the third area which is coming up is the complete new area of metabolic engineering or metabolomics, as it is called. So I have it somewhere over here. Yeah. So herein, by doing a lot of bioinformatics based background research, you can try to tweak not only one or two genes, but four to five important genes in a metabolic production pathway. So for this, you need to have a situation wherein the secondary metabolite pathway has been elucidated in greater detail, so that you know ki out of these so many 15-20 genes, which are the most important genes that can be manipulated. So some of you might be aware that in case of Catharanthus roseus, which produces the anti-cancer metabolites vincristine and vinblastine, people have identified the biosynthetic pathway to a great extent, and many labs all over the world are working towards the area of metabolic engineering by manipulating the crucial genes in the vincristine and vinblastine biosynthesis. So, three to four genes are manipulated simultaneously when you are concentrating on metabolic engineering. Whereas when you are looking at genetic transformation through agrobacterium, you are trying to change only one or two genes. So that is roughly the distinguishing main distinguishing factors between these two methods. But again, as I told you, I will not be covering this in my talk in detail. So these are a few of the examples which are very well-known examples in literature, wherein by use of tissue culture methodologies, the production of the important secondary metabolite has been upregulated to a greater extent as compared to intact plants. So you can look at this right-hand side column, which tells you the amount of the secondary metabolite which is present in the mother plant. And here you can see that through tissue culture, it has increased to a very high extent. So as an example, if you look at these gallium anthraquinones, they were present at such a low value. And here you can see that straightway there is a jump of at least 10 times to even 20 times, and so on and so forth, in this increased value via tissue culture. So there are many such examples. In Lithospermum erythrorhizon, which was one of the first success stories which came from Japan, this yield increase has been tremendous. So you can see here it is 1.5, and in the best culture medium it is 23. So you can see that there is 23 times increase via simple tissue culture. So no, nothing like agrobacterium mediated transformation, etc., etc. So this chiconin production. So chiconin is a red colored dye. From the species name, you will realize erythrorhizon. So roots which give rise to red dye. So the root culture was the initial starting point for this red colored chiconin production, and this dye has got both cosmeceutical properties. So it is used in many of the traditional Japanese cosmetics, and also as a food ingredient it has been used. So I have subsequently also mentioned about this, wherein people have standardized huge bioreactor level cultivations for this lithospermum. And there are so many other examples, but as you will see, most of these examples are coming from the Japan, Korea, and China, and to some extent from Germany. So these are the leading players in this entire area, I would say, wherein the bioreactor technology for secondary metabolite production has been regularly used at a commercial level. Then there is this another criteria which people have applied, which means the novel secondary metabolites. So that means, what is meant by novel? So all of these secondary metabolites are not found in intact plant which is growing in the field. But when you bring these plants into culture, under the influence of culture conditions, under the influence of the plant growth regulators that you have used in culture, there are some additional branches that develop in the secondary metabolite pathway, and you get production of completely new metabolites in tissue culture. So obviously now these metabolites will have their own therapeutic activities which are completely novel from the intact plant. So this has been one of the most interesting aspects, that production of completely new secondary metabolites when you are working with in vitro cell cultures. Then again, there are so many parameters, because it is not so easy to increase the yield of these secondary metabolites to this much of a level. So at least 10 to 20 times increase in the yield is required to at least think of that methodology for application at a commercial scale, right? So based on that, the people will start to think ki whether now we can increase it to a, like 2 liter flask level. Then they will again study if it is increasing. Then they will take it to a 10-liter bioreactor, and then finally to 500 liters or 1000-liter bioreactor, which is normally used for, fungal cultures, wherein our many of the antibiotics are produced. So, as I was telling you during my visit to Korea, I was fortunate to see one of the labs wherein this 500-liter bioreactor was there, which was producing metabolites which were directly used in their cosmetic preparations. So obviously, since many of this research is protected under patents, a lot of details were not revealed. But the bioreactor culture, I was aware that it was from our Morinda citrifolia, which is called as noni, and it is a very important plant again in the Korean pharmacopoeia. And they have standardized bioreactor scale cultivation of Morinda cell culture. And the extract is directly used in cosmetic products by many leading cosmetic companies like Garnier, etc. So, lot of modifications can be carried out in all of these parameters. So, media variations, culture condition variations, then a number of specialized techniques. So all of these variations you have to standardize for each and every plant, so to say, because each plant has own personality. So depending upon that, you have to standardize all of these conditions, and then only the plant is going to give you a wonderful response. So these are the very well known cases wherein people have got extremely high productivity levels. So all of these again are coming from Japan and Korea, because they already have bioreactors in place for all of these. I'm sorry, there is a spelling mistake over here. It should be Coptis japonica. So berberine, again, in initial days, berberine was only thought to be effective as a cosmetic, but recently it has given extremely good results in diabetes trials also. So the clinical trials are completed, and people are now waiting for berberine to come out as a therapeutic for diabetes. It has shown very nice results, just like metformin. So rosmarinic acid, again, a very well-known compound that is having both therapeutic as well as cosmetic properties. Then two plants for that, Salvia officinalis and Coleus blumi, and then Taxus canadensis, obviously for the anti-cancer Taxol production. So this was the um, bioreactor that was initially used for the, so this shows the upscaling, how it happens. So initially it is upscaled in these small benchtop type bioreactors. Then it goes ahead and gradually the capacity of the bioreactors is increased. And here you will see that this is a 70,000 liter bioreactor. So it is actually from a company in Germany called as Phyton Biotech. Similarly, there is a company in Korea called as Samyang Genex, which are working with Taxus hairy roots. So from hairy roots, they are isolating Taxol, and they have also upscaled the hairy root production in very, very high values of bioreactors. Uh, Unfortunately for us, and fortunately for the companies, all of these details are patent protected. So you will never see publications coming out of it in open journals. So many of their improvements, what all media manipulations and all they are doing, how they are cultivating, cultivating plant cells in such a high volume of bioreactor is so very much tricky because of a lot of biochemical engineering constraints. But they have been able to do it, and successfully, because if it is not giving them good yields, the company is immediately going to dump that. But they are working on it since last at least 10 years, so that means it's a very, very robust process, and they are getting a lot of commercial value out of it. So this is from a latest review, wherein I have been able to find out which all companies are working in this area. So as you can see, most of them are under Mitsui. So to say Mitsui from Japan is a petrochemical company, but they have a huge biotechnological arm also. So the profit that they get from petrochemical industry, a chunk of it is given to the biotechnological wing. And because of that, they have come out with so many successful molecules. All of these are cultivated in bioreactors. So chiconin, berberine, as I told you, now berberine cost is going to increase like anything, because diabetic population is increasing all over the world like anything. And if berberine is now cleared all the clinical trials, so berberine is coming out in the market in next couple of years, I would say. And then this company is again going to make a lot of money based on this. Then ginsenosides. So a lot of ginseng products are high in demand in all of these Southeast Asian countries. So they have their native demand itself is very, very high, which is because ginseng is almost extinct in its natural habitat. So whatever Panax ginseng is there. It is under cultivated gardens or in tissue culture. So people are working on that. This Nippon Denki, again a Japanese company. So here you will see that there are only two German companies which are uh, successfully marketed these drugs from the bioreactor level. All others are from Japan and a few are from Korea. Those are not mentioned in this table because I have taken only a small part of the entire table from the review. Okay, so I'm not going to talk about the transformed cell cultures because it is again a completely different area. But yes, I would like to talk about this relatively recent area around 10-year-old, which is called as molecular farming. So here you will see that how the name has come. So it is pharmaceutical. So FARMING instead of that, it is PHARMING. So what is done is that the plants are transformed with pharmaceutically important molecules. So one or two genes only are changed, and then you grow the plants like a agricultural crop. So here there is no intervention of bioreactor technology, etc. You have a transformed plant in your hand, which is normally a fruit crop or our cereal crop. Those are the successes which are reported, and then you cultivate them normally like your normal farming practices. So obviously all sorts of clearances, environmental clearances, ethical clearances, etc., have to be in place before this can be done. So obviously in India, none of this research is done because India is still not permitting a lot of trials for genetically modified crops. But in Western countries, these have been routinely done. And then again, one example from Japan I'm going to show you in my subsequent slides. So here, so many properties people have tried to improve. So you have therapeutical products, nutritional components, this vaccine antigens. So it is, you might have read about it. It is called as edible vaccines. So rather than taking an injection and then going in again, remembering the next dose dates, and again going to the clinic, etc., people have express the antibodies in the plants effective against a number of viruses, and then they say that you can directly ingest these plants or the fruits or whatever product is desired, and then you will get your dose of the vaccine. Then biodegradable plastics and many industrial products. So all of this comes under the purview of using the whole plants as bioreactors. So here you are just transforming the plants and then growing them out into the field. So again, I'm not expecting you to read this, but I'm just want to emphasize the point that this area is so attractive that so many companies all over the world are interested in this. So Ventria Bioscience is a leading one in this. You can look at their website and it will give you a lot of information. Then Planet Biotechnology, but it is working with tobacco. So they are working in expression of vaccines, so-called vaccines into the tobacco leaves. Um, but the crop plants, then a lot of other examples are there. So I have just made a slide related to this. So in banana, no, banana is an attractive species to work with because you can eat it raw. For example, if you are wanting to like expression in cereals, so in expression in rice as it is shown over here, or in maize, so you cannot use the grains in their raw format, they need to be cooked. And as you know, many of these antibodies are proteinaceous, so there is a danger that those can get degraded during the cooking process. So people are working on these raw fruits which can be eaten raw. So banana can be eaten raw, tomato can be eaten raw. So the idea is that when you eat as per your body weight, the dosage will be calculated. and you will be asked to eat either 2 or 3 bananas. The expression levels are quite high in these foods. So there are reports of successful production of hepatitis B vaccine. So jaundice or कावीळ ज्याला आपण म्हणतो. So त्याच्यासाठीच अँटीबॉडी इथं प्रोड्यूस केलेली आहे. In tomato, you have anthrax and rabies vaccine, which is getting expressed. So the proteins are getting expressed in all of these plants. This study is from the Tokyo University in Japan, and they have made this edible cholera vaccine for kids, because normally even in India we give the semi-cooked rice to children. Uh, as a staple food for them. So they say that you do not overcook the rice, but then it can be made into like a paise or a kheer, and then you can get the desired dosage of the anti-cholera vaccine through this genetically modified rice. This is a tobacco wherein anthrax and AIDS vaccine studies are going on. They are still not out in the market. They are undergoing clinical trials. So this is an interesting area which is again related to little bit of therapeutics. So I thought of including it over here. And now I will end my talk with an interesting idea which is not related to secondary metabolites, but I thought that I would share it with this as a fun fact. So there is this entire area of 3D printing of plant tissues for food manufacturing, and you can directly, so this is a spinach leaf which has been modified to be produced in the form of a dinosaur. So people are thinking of this as a way to interestingly produce the food items so that even people who are not interested in eating those, but because of the good properties present in the uh, in the like spinach leaves are extremely healthy and considered to be very good for our daily diet. So but many children are not interested in eating that. So if they can be made into this format, whether we can um, entice the children to eat this. So this is just the beginning. The first paper came out in 2017, and since then many companies have ventured into this area, and they are into from that review only I got these figures that they have, you can print a food in the form of a three-dimensional shape as well. So that is all from my slide. Uh, so I will be happy. Thank you.
From my side, ma'am, I want to just know what is scenario of secondary metabolite production in India when we compare it to other nations. Is it really developed in India or it is in developing condition?
So right now, as far as I know, there are only 1 or 2 companies who are seriously looking at least bioreactor level standardization. So BARC had developed some indigenous bioreactors when Dr. Hebre was there. And from there, actually, some people had tried to work on it. But as far as I know, only Dabur had reached to some level of success in terms of Taxol production, because they were interested in hyper production. But other than that, up to my knowledge, nobody has taken it to a commercial level as yet.
How many can say types of product they might have tried? I don't know exactly the uh, plants which are tried for secondary metabolite production because there are many, I think, plants which might have tried, but from India, which plants they are tried for secondary metabolite production under tissue culture? Most probably, maybe can say through callus or through hairy root culture.
So research articles are in numbers. So from
Hmm.
India, a lot of research articles have come out for secondary metabolite production. But as I told you, commercial level production, I don't think so. At least it has not been reported. Key
Okay,
people
that's
have
it.
successfully upregulated it to that
Mhm.
much of a level, because in order to take it even to bioreactor level, As I
Mhm.
showed in that table, initially you need to have at least 10 to 20 times yield in your, flask culture.
Mhm.
And then only you can think of taking it up further. So nobody has reached up to bioreactor level. Only BARC was successful in Coleus bloomy. They were
Mhm.
working on Coleus bloomy. But presently, I don't think so. Even they have any ongoing bioreactor studies. And that also, I think, as far as my knowledge goes, it was up to 10 liters bioreactor, not beyond that.
That means there is much scope for, can say, for this particular study and production of secondary metabolite in
Yes,
India.
actually, it needs to be developed, but somehow I feel that one thing is that even if you produce it through bioreactor, na, you have to again go through all the regulatory ways. Like, for example, again, Taxol. I keep on taking that example because I have read so much about it. Whatever clinical trials have been done, they have been done on Taxol isolated from the native plant. Now, if you say that you have produced Taxol in bioreactor, then again you have to go through all the regulatory trials to prove that your tissue culture or cell culture produced Taxol is able to satisfy all the norms, and the costing is enormous in such case. So that is why, because we are
Uh,
so much
I think,
lacking in funds, that is one of the major reasons why people are not coming into picture in this case.
what do you think students should go for, can say, this type of studies, specifically for their PhD work? Or they should take, can say, a tissue culture and production of secondary metabolite for their, can say, entire life as a cancer research area, and which are the probable plants where we can have, can say, much scope, or can say most, can say, prospective plants for this.
So again, I will not be able to pinpoint the plants per se, but if I would think of it from the other way around. So, which
Mhm.
are the two major diseases that are, or the three major problems that are currently there? One is obviously cancer, which is not going to go away. So, anti studies of anti-cancer plants. But again, I have seen many PhD theses also come for reviews, etc. People
Mhm.
will just do a basic MTT assay and they will stop at that. So that is
Mhm.
not it. You have to understand exactly what is happening further. So MTT is a general cytotoxicity assay. So you have
Mhm.
to go further and identify the target on which it is acting. So like Taxol acts on tubulin. Then
Mhm.
currently I'm working on two Western Ghats plants. They are working on apoptosis. So
Mhm.
likewise, you have to go ahead and show that this is going to act on this particular target. Then it will have much more value. Sadly, people will just do one MTT assay and they will be
Mhm.
satisfied that my plant is having anti-cancer properties. But if you look at the global scenario, nobody gives any importance to a single MTT assay, and that's why the research papers also are not published in good journals. So you have to, MTT is just one basic preliminary assay. You have to go beyond that. Similarly, this antimicrobial resistance, the development of resistance to the presently available antibiotics is so strong that really we need to find out many more effective molecules. But again, just doing a disc inhibition assay is just not good enough. So you have to move beyond that. So that is the thing that I want to emphasize. Diabetes again is not going to go away. You must have seen the recent ICMR report. Literally every fifth person is having diabetes in the adult age group, and the age of onset is decreasing every year. So if people who are hardly 33, 34, they are getting detected with type 2 diabetes. So these are some of the diseases which can be targeted by people. And since in India, many of the plants are so much understudied, we don't know exactly. From Western Ghats, there are so many underexplored plants. We just don't know against which disease they are active. So, such kinds of exploratory studies definitely can be taken up by students. Some very rare plant. So, because rare plant will have good rationale for taking it up in tissue culture. And then propagation of that as well as its therapeutic activity elucidation will be a good way in which students can move ahead.
That's nice. Uh, I think some of the students are working on medicinal plants, some of them are
Okay.
working on physiology of the plants,
Acha.
and some they have studied, I think, microbial activities.
Hmm,
No,
hmm,
so
hmm.
this is a versatile group, and
Okay.
definitely they will benefited out of your lecture. And you are given the brief idea what secondary metabolites are and how we can produce it, and what is the largest site of, can say, size of bioreactor. I think Japan, you are
Hmm.
given the example. First time I heard about this is the size of bioreactor where you can produce Texel and all. That's nice. But I think there is much scope for the students, and there is what can say huge room for the development of the studies.
Yes, yes.
Thank you very much, ma'am.
Thank
Uh,
you.